Okay, so I think we'll get started. So first of all, I just want to remind everyone that the session is being recorded. Welcome to all to the inaugural AI healthcare rounds at McMaster. This is a new initiative that we just started, and it's really meant to be a multidisciplinary, collaborative kind of forum for people to bring forward new AI ideas, research that they're working on, and to really help share and generate new ideas and perhaps even get some input from people from other fields. We have a large variety of people on the call today. So, we have physicians like myself, scientists, including from biochemistry and computer sciences, engineers. We have folks from industry, students, and I'm sure I'm missing a bunch of others. So I'd really like to welcome everyone today. Just about myself briefly, I'm an abdominal radiologist at Hamilton Health Sciences. I'm based out of the Juravinski, with an interest in the AI space. And we have Dr. Stokes presenting today, who has had quite a bit of success for someone at his stage of his career. So just to give you some background on Dr. Stokes, he did a Bachelor of Health Science and PhD here at McMaster University. He did a postdoctoral fellowship at the Broad Institute of MIT and Harvard from 2017 to 2021. And then he established his lab at McMaster in August 2021. He has an interest in experimental and computational approaches to discover the next generation of life-saving antibiotics, and has a focus in particular on deep learning approaches to predict the properties of structurally novel small molecules. And with that, I'll hand it over to Dr. Stokes, who will be discussing AI in antibiotic discovery. I should actually mention one other thing. Sorry, John. If people have questions or comments, we want to encourage people to speak up even during the presentation. I think it might be structured a bit didactic, but we really want to encourage conversation. And so please feel free to raise your hand or start speaking during Dr. Stokes' presentation.
Great, thanks, Christian. You can see the proper screen and everything. We're good to go. Awesome. So I'm John. I'm an assistant professor in biochemistry. I'm also a faculty member at the Hearsing School. And like Christian said, I'm going to be talking about the application of AI in new antibiotic discovery. Also, like Christian said, don't hesitate to ask questions along the way. Today, I'm more interested in teaching something than getting through, a nice, tidy story. So Christian will be monitoring the chat. I can't see anything on my end, so I'm just going to rely on Christian to chime in, but you can all as well. Okay, so here's why I do what I do. So here's a timeline of antibiotic discovery. You can see we've had antibiotics for the better part of about 100 years now. But what you'll notice is, nearly all of our current clinical classes of antibiotics were discovered between the 1940s and the 1960s. This is the so-called golden era of antibiotic discovery. And it's a period of time characterized by, high productivity in discovering different structural and indeed functional classes of antibiotics. And this was done largely through screening secondary metabolites produced by microbes that live in soil. So microbes that live in dirt, environmental conditions, have evolved mechanisms to make their own antibiotics as a form of microbial warfare to compete for limited resources in various ecological niches. So, in the middle of the 1940s, we figured out a good way to mine secondary metabolites for new clinical antibiotics, and we got really good at it. You can see, however, come the middle of the 1960s, our ability to discover new clinical antibiotics through this approach of identifying secondary metabolites from microbes decreased. And the reason it decreased is because we kept rediscovering the same antibiotics over and over and over again. This is something that is known as the dereplication problem, or the rediscovery problem, depending on who you talk to. So what happened, in more recent decades is we've shifted away from natural product mining, more towards synthetic small molecule screening. So what this looks like is we'll go into the laboratory and we'll physically curate hundreds of thousands to maybe millions of molecules at the largest scale. And then we'll take those small molecules, those synthetic small molecules from our freezer, and then we'll test them against a microbe of interest. Unfortunately, over numerous decades, and I don't know how many millions of molecules that have been screened, we have discovered precisely 0 new clinical antibiotics through this approach. Now, it's important to note, we can be a little pedantic and think, okay, if our lean antibiotic discovery pipeline existed in a vacuum, we'd still be okay because we have a whole bunch of antibiotics. But our lean antibiotic pipeline does not exist in isolation. Indeed, what coincides with our lean antibiotic pipeline is the global dissemination of antibiotic resistance. It's been, a couple papers came out recently. It's been guesstimated that a couple million people died in 2019 due to drug-resistant infections. And that number is expected to expand to roughly 10 million people per year by the year 2050. That mortality exceeds that of cancer. So the question becomes, how can we repopulate our lean antibiotic pipeline with new potential clinical antibiotics? And I am a proponent of the use of artificial intelligence in that endeavor. And I'll explain why. So if we look on the right-hand side of the screen here, this is what a conventional small molecule screen looks like. We'll curate, hundreds of thousands to maybe a couple million molecules. We'll screen it against a pathogen of interest. We'll get our hits, and then we'll go chase them in the laboratory, figure out what they do, their the positive attributes, negative attributes, et cetera. But this overall approach is limited, at least in my opinion, for 3 reasons. First is it's really expensive, right? Because if you want to screen a million molecules, you have to go buy a million molecules. 2, it's time consuming, because if you have a million molecules you want to test, it means you have to run a million individual experiments, right? And 3, we often find that the chemical space that's explored in these chemical libraries is not as vast as we would hope, right? So we're not actually getting a good exploration of the totality of chemical space. So we can now compare that to what I'll call a machine learning guided approach to antibiotic discovery. And the process looks a little bit different. So we'll first go into the laboratory and gather a training data set. So this is real data where we take a handful of chemicals and test them against a bug of interest, let's say. And here, instead of having to run, hundreds of thousands to millions of experiments, we can get away with thousands to tens of thousands. So then we'll take that training data set, we'll train some machine learning model. And then once we're confident with the performance, the in the computer in silico performance of that model, we can then take that model and apply it to huge chemical spaces in silico in the computer. Meaning that once we have trained our model to understand chemical features of antibiotics, we can run predictions against new chemicals for new antibiotics in the computer at scales of, hundreds of millions or more. So you can see across those 3 dimensions I discussed, we see improvements. So first, it's a significant cost savings, because instead of having to screen a million molecules, we can screen 10,000 maybe. 2, in terms of time, we can run predictions in silico orders of magnitude faster than we can perform experiments in the laboratory. And 3, because we can perform predictions in silico so much faster than we can perform experiments in the laboratory, it affords us the opportunity to explore truly vast chemical spaces for new drugs. So it's sort of with this, theory that we always enter new antibiotic drug discovery programs with. So the story I'm going to talk about today is one that we recently published. It came out nearly 2 months ago, where we wanted to leverage machine learning to help us discover new antibiotics against this pathogen called Acinetobacter baumannii. So A. baumannii is a really challenging pathogen. It's a hospital-acquired bug, and it likes to live on, medical surfaces, so the doorknobs and medical equipment and stuff. And it's particularly burdensome because it likes to acquire extracellular DNA and just hang on to it. And you'd imagine that if you're a bacterium living in a hospital, a lot of that extracellular DNA is going to encode antibiotic resistance genes. So it's becoming increasingly common to find Acinetobacter infections that are resistant to most, if not all, currently used clinical antibiotics. Moreover, Acinetobacter can cause quite a diverse array of infections, things from pneumonia, so lung infections, to meningitis, to blood infections, bacteremia. They're common wound pathogens as well. So they cause a diverse array of infections, and they're multi-drug resistant. And recent estimates put mortality at roughly 1 in 4. So it has a really high mortality rate. So given that Acinetobacter is a global challenge, we figured, can we leverage ML to help us find new drugs against Acinetobacter? Okay, so like I mentioned, the first thing that we have to do is gather a training data set, right? So what I'm showing you here is data that we acquired in the laboratory. So we screened roughly 7,500 diverse molecules for those that inhibited the growth of Acinetobacter baumannii in a dish. So what we're looking at here, on the vertical axis, we see growth. And on the horizontal axis, we see all 7,500 molecules that we screened. Every dot is a different chemical. All right, so we have these screening data. And what we wanted to do is separate them into molecules that we defined as antibacterial and those that we defined as not antibacterial. So here in blue, we see all the molecules that we defined as not antibacterial based on some statistical threshold. And everything in red are molecules that we defined to be antibacterial against this bug in this experimental context, right? So what we did here, this is called binarizing the data. Either it did inhibit growth or it didn't. Okay, so what do we have? Well, now looking at this data set, we now have 7,500 structures, right? We know the chemical structure of every molecule that we screened in this data set. So now we have structure property pairs, right? If it didn't inhibit the growth of Acinetobacter in a dish, we labeled it with a 0. If it did inhibit the growth of Acinetobacter in a dish, we labeled it 1, right? So now what we have is a data set that we can now use to train a model, a machine learning model, to understand chemical features associated with these two properties, either growth inhibitory or not, right? So there's a whole bunch of different ways that we can do this. But we chose to use something called a message passing neural network. I'm not going to go into the details of message passing right now. We can later if you want to, but we don't need to right now. But at a very high level, what this type of model does is takes as input the structures of chemicals, right? So atomic interconnectivity of molecules. This is called the graph structure. So in machine learning, anything that's connected like a molecule where you have nodes, which are atoms, and edges, which are bonds, this is called a graph. So we take as input molecular graphs, and then through this message passing neural network approach, we can convert the structures of molecules into dense continuous vector representations. So we take the structure of chemicals and then convert it into a long string of numbers. And that's what's being represented here. A long string of numbers that captures the molecular features of the molecule. What happens then is this dense continuous vector that was computed during message passing then serves as the input layer to this thing that I've shown here. This is a typical feed-forward neural network, nothing fancy. So we have our input layer, and then data gets processed in the forward direction, represented by this arrow. And then the output of this feed-forward network is the prediction score, right? So what the model at a high level is doing is looking at the structures of molecules, learning the molecular features that either have antibacterial activity, meaning the output score would, or the prediction score would be close to 1, or do not have activity, in which case the model would be learning those features and the prediction score would be close to 0. Okay, so to reiterate, what we've trained our model on are 7,500 structure property pairs. And during training, the model is learning to extract relevant structural features of chemicals in the context of their associated property. And then what we can do is we can start to show this model brand new chemicals it had never seen before, molecules that are different from those during training. And based on what the model learned during training is it will be able to look at the structure of the chemical, identify potential chemical features that are associated with antibiotic activity or not, and output an associated prediction score. So here we have some brand new molecule the model didn't see during training. And then based on what it learned during training, it output a prediction score of 0.15, which is pretty low, right? Because remember, we told the model, if it's 0, it's not antibacterial. If it's 1, it is. So we're looking for molecules with prediction scores that are closer to 1. Okay, so this type of model is called a discriminative model. The model is discriminating between different molecules that we show it. And, you know, based on our ongoing work and our previous work, we can perform molecular property predictions for new antibiotic on the scale of roughly tens of millions per day, give or take. Oh, I see Isaac put a hand up.
Now that it's reached the point where I'm getting interested and excited about these molecules, I have a question. Okay, so you gave it a 0 or 1. So 1 inhibits, 0 doesn't. That's based on a cutoff that you decided, but really in the 1 category where it inhibits, it's a range.
Yep.
Is this for the simplicity of this model? It has to be binary, or can you actually create a model that is not binary and actually tells you this one's the best inhibitor, it gets a 1. This one is 80% inhibitor, so it gets a 0.8. And then if that's possible, and you'll tell us, would it be able to predict better molecules rather than to give you a molecule that says this might work because it's in the range of 0.5 to 1? I don't know if that makes sense.
No, that's like the perfect question. So what I described here is something called a binary classifier, and we did it because we didn't have enough training data to train the model that you just suggested. And I'm going to be honest, the model that you just suggested is what we're working towards right now. It's just a matter of how much training data we can acquire. So the model that you just described is something called a regressor, right? It's a regression task as opposed to a binary classification task. So in a regression, in a regressor, what it's trying to predict is how much inhibition are we going to get with a specific structure,
right?
Not will we get inhibition, yes or no. But you can imagine, hang on, I'm going to go back a little bit. You can see my cursor, right?
Yeah.
All right, so see all in the middle, like kind of like in this gray zone here, it's not very densely populated with data. Right. So that because we don't have a lot of examples of molecules that kind of inhibit in like this like gray zone range right here, the model failed miserably when it was trying to predict molecules within that range.
Right.
And we have like empirical in silico performance data to support that. So we, instead of trying to train a regressor, we just said, okay, let's just do a binary classifier for now. And over time, we're always continuously retraining our models with additional training data. And we can see that over time, we can populate this region of growth inhibition with a diverse array of structures. And now, like, we're just starting to get into a position where training a regressor makes sense.
Okay. And does the success of a regressor rely on the how big the original library? Like you started off with 7,500. If you started off with 75,000, are you going to end up with a better model? Or it doesn't matter the size of the initial library, it's the density in that middle zone that you really need to be big.
Yep, and I think there's a positive correlation between those two. As you screen more molecules, the probability of getting molecules that exist along this gradient from 0 to 1 increases, right? So I don't know how many molecules we'll have to get up to. Right now, we're probably at about 30,000-ish, give or take, and we're starting to like get the very, very beginnings of a reasonable regressor. But I think you're in the right ballpark. It's probably around 100,000 molecules before you can train a robust regressor. that makes predictions where it's like, okay, at some defined concentration, 10 micromolar, let's say, this molecule that you showed is likely to inhibit 50%.
Perfect. And last question is, when you say that 7,500 or the 100,000 screen, that screen has to be done in vitro, or is it an in silico, or both, or where do you get the data from?
Yeah, so we always go into the wet lab and gather our own data. Strictly speaking, you don't have to. Like, you can take data that previous groups have published, right? Associated with their papers for whatever, drug discovery task in which they were interested. The reason that we always go into the lab and gather our own data for a specific model, let's say, is because we have complete control over all of the experimental variables. So, there's always a little bit of noise associated with training these models anyway. So we want to have as much control over the input data as we can, so we can make the best decisions when a model makes predictions for us, right? And it kind of gives us after a little bit of practice, we can get a feel for, when the model is going slightly astray. Even if the in silico performance metrics are good, we can look at the output structures of compounds and say, I don't like what it's doing. I'm going to go tune my model a little bit more to move it to be able to predict one different type of chemical over another, let's say. And that's really, in my opinion, only possible when you have, when you've actually gone into the laboratory and gathered your own training data yourself.
Excellent, John. Amazing. Thanks.
Does that make sense? Okay. Oh, I see.
Absolutely. Absolutely.
Mahan. Sorry if I'm not pronouncing your name right.
Yeah, that was correct, actually. So sorry, John, could you please go to the next slide where you have yeah, that's it. So my question is specifically regarding how are you going to quantize the molecule so that you can feed it to the network? Because I work with images, and in images, let's say your image is 50 by 50, and then you have 2,500 pixels, and let's say each pixel is mapped into a 8-bit integer, and then you have a number for it. But because I'm just new to these antibiotics and these stuff, I don't know how you're going to quantize your molecule and then feed it to the network. So this is my first question, and then the other one is depending on the structure of the molecule, how big or how many neurons in the input layer do you have, so that, for example, I don't know, one molecule may not be really large, so that maybe 10 neurons are enough to take as input whatever data that's from the molecule. But in some cases, maybe your molecule is so big, and you need to the number of neurons in the first layers should be so large.
Yep. Okay. So I'm gonna just skip to a slide where I actually have this. So you can see my slide, right?
Yep.
Okay. So I'm just going to discuss how this sort of message passing paradigm works at a high level. And it'll be very clear how we actually develop that input layer to the feed forward network. Okay. So we see this molecule here. This is the exact same one that I showed on the previous slide. So how we start is every bond in a molecule is given an initialization vector. And this initialization vector is generally, this is a hyperparameter that we can tune, but it generally sits between 300 and 1,000 dimensions, something like that. And every bit in the vector is when you initialize that vector is computed using actual atomic and bond properties. So we initialize every bond in atom with an initialization vector. So, and then we do message passing. So let me explain it. So see this bond, here's some bond in the molecule, we labeled it 2,1. It is represented just for visualization purposes by this 3-dimensional vector here. In reality, let's say this is a 300-dimensional vector, right? So what we want to do, if we're moving left to right across the molecule, we want to look at all of the bonds that feed into that bond on which we're focused. So here we're looking at 2,1. Well, we can see that 3,2 and 4,2 feed in to 2,1. Now, 3,2 and 4.2, they both have their own vector representations, right? Here it's a 3-dimensional vector representation. In reality, it's 300. Okay, so what we do in order to inform bond 2.1 about its local molecular neighborhood is we sum 3.2 and 4.2 together. And then what we do is we take that summed vector and we concatenate it onto the original vector that represents 2.1, right? So then we have this, I'll call it like 2 n-dimensional vector, this concatenated vector that then gets pushed through its own feed-forward neural network to result in an updated n-dimensional vector, 3-dimensional or 300-dimensional vector that represents an updated state of 2.1. And this updated state has information now about 3.2 and 4.2. So during message passing, what we're doing are transferring information, if we want to consider that, across bonds of a molecule. And then what we do at the end, what we'll do is we'll do maybe like between 3 and 5 rounds of message passing. And then at the end, we take all of the updated bond representations and we just sum them together to end up with our, our single dense vector representation of a molecule. And then that dense representation is what serves as the input layer to this feed forward network. Does that make sense? Does that sort of capture how we're, now this isn't the only way to do it. This message passing approach just tends to work a little bit better than other more simple approaches for molecular property prediction tests, but there are 100 ways to do this.
Yeah, that completely makes sense. Thank you so much.
Awesome. Cool. Molecules and predictions. Okay, so once we trained our model on the 7,500 structure property pairs, we ran predictions. So we actually went to a relatively small chemical data set for our initial set of predictions. We used the drug repurposing hub, which is a drug repurposing library at the Broad Institute. And so what we did is we took all the molecules from the drug repurposing hub and just showed them one by one to our trained model, right? So we were showing them the trained model pictures, for lack of a better word, of all the molecules in the drug repurposing hub. And this is what that data looks like. So what we're looking at here is prediction score. So this is what the model thought, between 0 and 1, all these molecules were antibiotics or not. And then we just ranked all the compounds from lowest to highest. And you can see if we look at the distribution of prediction scores, a relatively small number have high prediction scores, but most of them don't. And that kind of makes sense, right? We know that the universe is not filled with antibiotics, so only some of them are antibacterial, and this sort of makes sense. It passes like the gut check, right? So we looked at the distribution of prediction scores and, quite practically just said everything above 0.2 seems like a good number, right? So we defined everything with a prediction score above 0.2 to be potential antibiotics based on prediction scores from our model. But you have to think, just having a good prediction score isn't enough. When we're looking for new antibiotics, we're looking for structures that are fundamentally different from all of the known clinical antibiotics we have. And that's for 2 reasons. One, if we have structural divergence in new antibiotics, the probability of being susceptible to pre-existing resistance is low, right? And 2, as soon as you get into new structural space in the context of chemicals, it increases the likelihood that you're going to have a new biological function, right? So what we did is we said, out of all the molecules that have prediction scores above 0.2, which ones are structurally divergent from known antibiotics, right? And that's kind of what we're looking at here. So everything in blue this is a 2-dimensional representation of chemical space. Everything in blue is our training data set. Everything in red is our prediction set. And then we settled on this one molecule that we named abison, which was both structurally novel and predicted to be antibacterial by our model. Okay, so here's the structure of the molecule. It doesn't look like any known antibiotic that's existed. When we tested this molecule against Acinetobacter in vitro, so in a dish, we observed that it had a minimum inhibitory concentration of about 2 micrograms per mil, 2 to 4, depending on the day. And what we're looking at here is a plot of growth as a function of drug concentration. And we can see, 2 micrograms per mil inhibits growth. And this puts it roughly in the ballpark of existing clinical antibiotics that are used to treat Acinetobacter. So that was check. What we then asked was, is this molecule bactericidal or bacteriostatic? Does it kill Acinetobacter or does it just inhibit its growth? We found that it's kind of in the middle. It doesn't completely sterilize cultures of Acinetobacter, as we can see here, but it isn't completely static either. It kills a log in a bit, and it sort of falls in the middle. Okay, I should explain actually on the vertical axis, what we're looking at is bacterial viability, so life, death. And here is our drug concentration. So we can see at higher concentrations for prolonged durations, we kill, 90%, 95% of the bacteria in that culture. Okay, this is when we got excited. We then asked the question, what is the spectrum of coverage of this molecule? Meaning, what are all the different bacterial species that we can incubate with this molecule in a dish and observe growth inhibition? Okay, so if we want to look at the top left, this is a panel of, I believe, 43 clinical isolates of Acinetobacter baumannii, representing an array of multi-drug and extensively drug-resistant clinical isolates. And we can see that as we titrate in more drug, we can eventually inhibit the growth of 100% of this panel. Okay, cool. But we ran an experiment where we started to look at the in vitro efficacy of abison against carbapenem-resistant Enterobacteriaceae, Pseudomonas aeruginosa, and Staph aureus, other problematic clinical pathogens. And what we observed was that even at very, very high concentrations, 20 times the MIC, we weren't observing any activity. And this was really weird because the overwhelming majority of clinical antibiotics are what are known as broad spectrum, meaning they are antibacterial against a wide phylogenetic array of bacterial pathogens. What we're looking at here, what this data initially suggested, is that abison was a narrow spectrum antibiotic. It had preferential activity only against Acinetobacter. And that to us was super weird and quite exciting, actually, because first, since we don't have a lot of antibiotics that are narrow spectrum, it automatically suggested that the biological function of this molecule was novel. But also, in a perfect world, all else being equal, I'm going to go out on a limb and say that narrow spectrum antibiotics are preferable for two reasons. One, broad spectrum antibiotics impose a universal selective pressure across an entire ecological niche. And what that means is that there is a strong selective pressure for antibiotic resistance genes to move really rapidly between species across that niche. However, a narrow spectrum drug, if it only targets one pathogen, the probability of having a high rate of horizontal dissemination of some resistance gene that will inevitably evolve against this is quite low, right? Two, we know that broad spectrum antibiotics, when they're given to patients, induce something called dysbiosis. It means the drug probably treats the infection, but it also will wipe out their entire microbiota, all the beneficial microbes that live in your gut. And that's associated with a whole bunch of deleterious side effects as well. For those two reasons, one, microbiome sparing, two, decreasing the rate of dissemination of resistance, I believe that narrow spectrum molecules are preferable to broad spectrum agents. Okay, but this was just part of the story, right? All this data says is abison doesn't kill other pathogens. But what we really care about is human commensal species. Does abison spare the human microbiota? Or does it have the potential to spare the human microbiota? So what we're looking at here is a heat map where red is dead. If you see red, that means the bugs are dead. So what we did is we gathered a collection of human gut commensal isolates and human skin commensal isolates. And we tested abison at 4 concentrations, so 0, 1, 10, 20x MIC. And then we tested 2 known broad spectrum antibiotics. A is ampicillin and C is ciprofloxacin. And what do we see? Well, abison doesn't really, kill a lot of stuff, as we would expect based on the previous slide's data. Ampicillin is a broad spectrum molecule, so it kills a lot, and ciprofloxacin is a broad spectrum molecule, and it also kills a lot. What I'll draw your attention to here is that at 10x and 20x, the MIC, so high concentrations of abison, we actually get growth inhibition against bifidobacteria. Bifidos are phylogenetically distinct from Acinetobacter. I don't know how many, hundreds of millions of years of evolution they're separated by, but it's a lot. These are gram-positives. And we still don't understand the biological function of abison against bifidos. It's complete, we don't understand it. But it's something that we're trying to avoid as we're working on making abison better. But anyway, this was at very high concentration. So we said, okay, it might have some, low level activity against bifidos, but overall, this molecule that we discovered is, for all intents and purposes, Acinetobacter specific. And that raised a really, really interesting question I alluded to earlier, right? And that question is, well, what is the biological function of this molecule? What's it doing? It's doing something new, but what is it? So we embarked on this project now to understand the biological function of abison. And so the first thing that we did in order to begin to understand the function of abison against Acinetobacter is, ironically enough, evolve resistance against it. So what we did is we take just wild type sensitive Acinetobacter, bathe it in the compound for a while, and over time we will observe resistance in the laboratory. This is all done on a dish. And then what we can do is we can take those resistant strains, do whole genome sequencing, find the position of the mutation that is conferring resistance, and oftentimes, not always, but oftentimes, that mutation will map to the biological target of the molecule. And so that's a way to help us understand the biological function. So we did this experiment, and we got 4 independent isolates that were resistant to abison, and they had mutations A362T, Y394F, and an intergenic mutation. And all of these mutations were in or upstream of the gene encoding LOL-E, this guy right here. So LOL-E is an essential protein that plays a role in lipoprotein trafficking. What does that mean? Well, bacteria make proteins on the inside of the cell, and then they have to ship them to the outside of the cell sometimes. Well, this protein, LOL-E, is an essential member of a whole bunch of proteins that facilitate that transfer of lipoproteins from the inside of the cell to the outside of the cell where they perform their native biological function. Okay, cool. Well, because we had a mutation in a protein associated with the cell envelope, we wanted to make sure that this mutation was specific. We said, does this mutation confer resistance to other antibiotics? Because if it did, it wouldn't help us understand the biological function of abison, right? So what we're looking at here are just abison-resistant mutants. 1, 2, 3, 4. Those 4 mutants from this slide. 1, 2, 3, 4. 1, 2, 3, 4. The 4 resistant mutants of abison and wild type, just normal Acinetobacter that isn't resistant. And then we tested it against a whole bunch of different structurally and functionally diverse antibiotics. And what we were looking for was to ensure that the resistant mutants that we evolved against abison were not cross-resistant to other antibiotic classes. And indeed, that's what we observed. Okay, cool. So this is good. It means we have a set of mutants that are only resistant to the thing that we're trying to figure out what it does. So then, before we started doing a whole bunch of technical laboratory work, we went back to the computer and said, if we look at the structure of LOL-E, of Acinetobacter, the protein in which we were seeing all these mutations, where does our most, I'll call it efficacious mutation lie? The one that causes the biggest increase in MIC, the one that is most resistant. Where does it live? Well, the structure of LOL-E for Acinetobacter hasn't been solved, so we predicted the structure using an algorithm called Rosetta Fold from David Baker's lab. And what we did is we overlaid the predicted structure of Acinetobacter LOL-E onto the known structure of the E. coli LOL-E, right? So in orange here, we see E. coli LOL-E overlaid on top of that is the predicted structure of Acinetobacter LOL-E. And the mutation that we were getting in Acinetobacter that resulted in the highest level of resistance was this A362T mutation, which is shown, represented by this blue blob here. Now, the homologous position in E. coli is I365, which is this blob right here. And what we're looking at here in gray is the acyl chain on the nascent lipoprotein. So what happens during lipoprotein transport is LAL-E kind of hangs out with the nascent lipoprotein as it's being shuttled. So this is part of that nascent lipoprotein. And what we see is our best mutation, our best mutant, has a mutation in like kind of the business part of the protein, right? That associates near the nascent acyl chain of the lipoprotein. And this strongly suggested that our mutation was doing something that was biologically interesting. So when we saw this, even though it's a prediction, we said, okay, this is worth really, investing heavily in. So we're going in now, and we have a strong mechanistic hypothesis. We think that abison is inhibiting lipoprotein trafficking via a mechanism involving LAL-E in some way. So the first thing that we did now was turn to RNA-seq. So we said, if we take abison and treat wild-type Acinetobacter in a dish, what is it trying to do? What is the transcriptional response? Is it consistent with inhibition of lipoprotein transport? And the answer to that question was yes. So when we took Acinetobacter and treated it with abison, we saw an immediate downregulation of genes involved in electron transport and transmembrane ion transport. And this transcriptomic signature is consistent with activation of this thing called the CPX membrane stress response, which is activated upon perturbations in lipoprotein trafficking in E. coli, right? So we had to lean on a little bit of E. coli literature and try to draw, conclusions in Acinetobacter, but there's enough conservation of these biological functions between these species that I think we're safe to make that guess. Okay, we then did a couple genetic experiments. So if we hypothesize that LAL-E is the target of this new molecule, we should be able to perform two experiments that help us to help gather additional data suggesting this is true. The first experiment we can do is we can take LAL-E and knock it down, make the cell make less LAL-E. And if we have a cell that makes less LAL-E, theoretically, we would need fewer molecules of abison to elicit a biological effect, right? And that's what we see here. If we take Acinetobacter, knock down LAL-E, abison becomes more potent. Check. Now we should also be able to do the reverse experiment. We should be able to overexpress LAL-E and observe resistance. Luckily, we didn't have to do any engineering for this. Nature did it for us. Remember I said we had an intergenic mutation? Well, this intergenic mutation resulted in an overexpression, a fourfold overexpression of LAL-E relative to the other mutants or wild type. And quite interestingly, this fourfold increase in overexpression of LAL-E in that mutant was well aligned with its fourfold increase in MIC. I'm not going to go so far as to say there's a true stoichiometric relationship here, but maybe. Okay. The last thing we did to elucidate mechanism was a microscopy-based experiment. So previous work out of Alita Miller's group has shown in E. coli that chemical inhibition of lipoprotein trafficking results in very specific, or I should say very clear morphological features. The first one is you lose native rod morphology, and the second one is you lose nucleoid condensation. So we asked, can we phenocopy this in Acinetobacter? So what we're looking at here from left to right is increasing concentrations of abison against wild-type Acinetobacter. So this is 0x MIC and this is 2x MIC. So no drug and then enough. And what we see as we increase concentration is Acinetobacter is losing its native morphology, right? We're changing shape from here to here. And we're also losing nucleoid condensation. And we can see that here because we have these nice little bits of DNA. And then over here, they're all dispersing gross, right? So this was just another level of data suggesting that this molecule was indeed inhibiting, lipoprotein trafficking. And this phenotype is consistent with all of our previous omics, genetic and genetic engineering data. And then the last thing we did was just to see if this molecule had any potential to work in a mouse model of infection. So Acinetobacter baumannii is a really problematic wound pathogen. You'll often see soldiers fighting in the Middle East when they get wounds, it'll get infected with multi-drug resistant strains of Acinetobacter. So we asked a very simple question. We have this structurally novel molecule that was predicted by an algorithm that has a new a new biological function relative to the existing repertoire of clinical antibiotics. Does it potentially have legs as a true clinical antibiotic? So what we did is on the dorsal surface of mice, so just on their back, we induced superficial wounds and infected with Acinetobacter. And then over time, over the course of a day, we treated topically with this molecule. So we formulated an ointment and we just put it on the wound periodically over a day. And we can see that mice that received abison had suppression of infection relative to mice that received simply a vehicle. Okay. So this is sort of a Cole's notes of this paper that just came out, a couple months ago, where we leveraged a neural network-based approach to discover a structurally and functionally new compound with narrow spectrum activity against Acinetobacter. So I encourage you go ahead and read it if you'd like.
I just need to ask you a question, kind of a more general question. We have a lot of trainees on the call. You've done an amazing job here, and thank you for that. And just when folks are trying to maybe, explore AI or come up with a new project, is there any advice you'd give them? Are there certain areas that are ripe for this type of work? Do you have a certain process you go through? Is it better to be an expert in one domain first and then go from there? Or is it better to not know much and kind of approach it that way?
I don't know. That's a good question. I mean, I don't know much about anything. So I feel like that's sort of like a good place to start. But for us, in the context of molecular property prediction, for whatever therapeutic domain, for us, the go, no-go decision is the availability of high quality training data with which to train these models, or the ability to acquire that high quality training data yourself. That's really about how far we think about it. If we can't get good training data, these models aren't going to do what you want them to do, at least these property predictors.
Yeah, that's been my experience in the radiology world, just getting that good ground truth, and a lot of it's not easy.
Yeah.
I think we should probably wrap up. I want to thank everyone. Thank you, Jon. This is great. And thank you everyone for joining. It was really good discussion and some good questions. I just want to remind folks that these rounds are part of the HearSync School, and were established to encourage dialogue, collaborations, and to advance the application of AI in healthcare at McMaster. This is a pilot project that we're doing for the next year. We're planning to hold these rounds monthly, the third Wednesday of each month. These rounds will be archived at the Mac Drive HearSync channel. Jon, if you want to say a few final words, but that's really it. We can wrap up.
We got a bunch of work on the go at the intersection of like drug development and antibiotics, and then, so keep an eye out. And if you're interested, reach out to me. We're always happy to help. And I don't actually do very much. This is the crew that does everything, and they're way better than me across every measurable dimension. Thanks to all the people who fund this work, and special thanks to collaborators who are helping us develop the SynthMol algorithm. And thank you all for showing up. You're awesome.
Wonderful. Thanks, Jon.